Fortunately or unfortunately, we're in the hotbed of where invasive insects occur and kind of wreak havoc in our forests. Yeah, the most obvious thing I think now, since we have so many pests influencing our forests, are the kind of dead and dying trees that people might see driving around or walking around their neighborhoods. You know, some of the impacts that are less obvious but probably have even larger impacts are, you know, how is the decline and loss of these trees based on these insects affecting the flora and fauna that inhabit those forests? How does the microenvironment that these trees create change and influence other processes like carbon flux or hydrology? This summer, I've seen some of the highest hemlock woolly adelgid density I've seen in like over 10 years. And so the white woolly material that you see here have all been secreted through the skin of an adult adelgid. They place themselves at the base of the hemlock needles, secrete this woolly substance, and then lay eggs. So every one of these woolly tufts is an adult that has laid probably 50 to 100 eggs. One of the first things we do is try to take a good inventory of what's in the forest now, because sometimes change can happen pretty rapidly. You know, when I first started studying hemlock, I was lucky if I could take some pictures, right? But with the advent of these new tools like LiDAR, which emits pulses of lasers, we can start looking at the subtle changes in forests and trees a little bit on a finer scale. We can also look at broader areas using satellites and aerial flyovers as another way to try to get at a better handle on forest health in general, and then how we might be able to use those tools to actually detect new infestations elsewhere.
All of my projects start with some sort of satellite imagery. My research looks mostly at how landscapes change and evolve through time. So it's not just getting a single picture, but almost like a movie of imagery and understanding how forms and functions change. And so a lot of my work is focused on characterizing patterns and processes and then associating them with different drivers. So with imagery, we have this awesome advantage of global coverage. And so it's not just about what happens at one specific site, but where we can connect that to other sites that have similar spatial, temporal, and spectral patterns emerging. So it's really interesting with, say, Lomantria dispae, you can really see when you're out in a forest and it's been defoliated, right? You've got little nubbins of leaves left on the trees. It will look like early spring, even though it might be the middle of July or August. But from an image perspective, it might just look like an early spring image. So when it comes to looking for pest damage, we rely a lot more on the time domain. And so we look for these signals in how an observation deviates from the other observations in a time series. And all of my pest-focused projects have some interesting spin on how we process a time series to make an estimate of how different a given observation is from its history. Maybe the most surprising thing as someone who wanted to think of remote sensing and product making as things with answers and like very concrete is maybe the philosophical challenge of change and monitoring change. When I first started a lot of this, I wanted to just map forests as forest. They are trees. And I was very lucky to have an ecologist involved with my graduate work who would say, well, what kind of trees? Why do I care about those trees versus those trees? And really getting to know places and build in this extra on the ground kind of perspective, like on what makes a place unique. And so when I design my products, I want to work with the people who are going to use them downstream.
People are really excited about trees and tree rings. They know the basics, that you can count and figure out how old the tree is. My main subject of study is understanding the long-term development of forests and how that interacts with climate change. So climate change today, in the future, but a lot of it in the past. I believe very strongly that we can learn a lot about how forests might change by understanding how they change in the past. We're getting to the finer analysis, the fine scale analysis now, is we want to know what does hemlock woolly adelgid do to the surviving trees once they start killing the hemlock. So we're in a forest right now, very much like the one in New Jersey that we're studying for this project. It's a mixed forest of hemlock, white pine, this large red oak. Hemlock woolly adelgid came to this forest in northern New Jersey in the mid-90s, we think. The trees started dying when I first was there in the early 2000s. We went back and sampled, we put in plots. So now we're diving deeper into individual trees within these plots, and we're going to try to look at the density of the forest around each tree and see how it responds to the climate. We are taking microcores, which are essentially really slices, thin wafer slices of trees, where we look at the growth of trees from week to week. Then the real hard work begins in the lab, and they take wafer thin slices and they look at, like, oh, 3 cells grew in the last 2 weeks, 4 cells grew in the last 2 weeks, and you can tell not only how much it grew, but how did they grow? What are the cell walls like? What are the the structure of the cells like from the beginning of the growing season right to the end? So what we first need to do to take a microcore out of a tree is we need to get to where the cells are actually growing. So I get a little block of the bark removed, and then I pull out this instrument called a tree fork. It looks like a small pen. It's very sharp here at the tip. You have to be very careful with the tip, like all increment bores. This is where we're going to extract the sample from. You put it up to the tree, and let's see if we can hit it well. Yep. That sounds pretty good. You pry it loose, you tap it back, and then you have a little sample of wood right here. And so that's the little increment, the little microcore, as they call it, the little increment core. We want to know, when do these trees grow? How do they grow in relation to each other? Some of the species that we sampled, shagbark hickory, black birch, I don't think they've ever been sampled in this way. Who knows how they're going to respond? We have ideas, we have hypotheses, but we're still learning how and when trees grow. So how are they going to respond to climate change is just a whole other thing. And we're going to let the trees teach us.